Well, if I can get your attention, really it gives me extreme pleasure to welcome you to this fascinating lecture this afternoon entitled Novel Physiology of Bone. We're all really looking forward to hear Dr. Gerard Carcenti's talk. He's truly a true pioneer and a distinguished NIH alumnus. He's going to explain to us once distant world of bone regulation and energy metabolism are in fact intimately connected. Dr. Carcenti earned his MD and his Doctor of Science from the University of Paris in 1984, and for the next 2 decades he labored in 2 NIH laboratories. He was first a visiting fellow in the molecular and cellular endocrinology branch, which is now, or what is now, NIDDK, and then was a visiting and postdoctoral fellow in the National Cancer Institute's Laboratory of Molecular Biology. In 1987, he abandoned us for Houston, Texas, which he called home for almost 2 decades. He taught medicine and pursued his growing interest in skeletal biology at the University of Texas MD Anderson Cancer Center, and later at Baylor College of Medicine as a professor in the Department of Molecular and Human Genetics. Dr. Carcenti is now the Paul A. Marks Professor and Chair of the Department of Genetics and Development at Columbia University, and his work is funded by multiple institutes at the NIH, including NIDDK, NIAMS, and NIDCR. Carcenti and his colleagues have overturned some long-standing assumptions about bone regulation and energy metabolism. Their work offers many promising paths for future studies. They have, for example, shown that the hormone leptin powerfully inhibits bone formation by shutting off serotonin synthesis in the brainstem. Now, serotonin is obviously mainly known as a neurotransmitter in bone, but it's produced in the duodenum, and when leptin serotonin pathway is turned on, appetite increases and new bone is formed. He's also shown that serotonin strongly inhibits bone formation in a way that's independent of the gene thought to control osteoblast. And then finally, certainly not finally, we're going to hear much more from him, but he's continuing to explore the hypothesis that bone mass and energy metabolism are co-regulated. And he's made striking discoveries about osteocalcin, an osteoblast-derived hormone. Osteocalcin, for example, he found promotes insulin production by the pancreas, and it also increases cell sensitivity to insulin and enhances energy expenditure. Bone, then, as it turns out, is truly an endocrine organ, and we're certainly glad that he was in NIDDK for a good portion of his career. These really exciting discoveries have profound implications for the treatment and disorders as diverse as diabetes, osteoporosis, traditionally regarded to be distinct yet unrelated entities. We now have many new avenues for exploration and development of potential therapies to increase bone mass and reverse insulin resistance on the basis of the work that he and his colleagues have been involved in. So really it is truly a distinct pleasure to give you Dr. Carcenti, who will tell us more about the interwoven system of bone and energy metabolism. Gerard.
Thank you very much, Chris. Thank you very much, Greg, for this very generous introduction. And what I would like to do today is to go over some of the projects and maybe more why we do what we do the way we do it, more than the results themselves, because the logic of the investigation, in fact, is more important than what we at the end found. So since 5 to 10 years, my lab is not working anymore on developmental biology, but only on physiology, taking the skeleton as a tool and only as a tool to really illustrate 2 principles of what I think are absolutely dominant principles of physiology. And unlike developmental biology, which has been a field driven since the last 25 years by the conservation of gene expression and gene function, when one speaks of vertebrate physiologies, you cannot really rely on fly or C. elegans to study it. If you study physiology in vertebrate, it has to be done with a vertebrate mouse animal or animal model. And I think it's important because this notion that there is a conservation of gene and of gene function is so dominant that we think that is true in developmental biology will be true in physiology, and it is not. The second principle of physiology, which I think is really quite important, is that for every physiological function, there is more than one organ that is involved. And this is a concept that was put forward in the 30s by a physiologist working at Harvard, L.J. Henderson, which is a concept of mutual dependence. So we know that for the control of blood glucose or blood pressure, there is more than pancreas or heart involved. But if we look at other physiological functions, more localized or restricted geographically, I would say, such as erythropoiesis, it's as much a function of bone marrow than it is a function of kidney. And we knew since 60 years that bone remodeling, the function I'm going to start from, is regulated by the gonads and 4 obscure glands in the neck, the parathyroid glands. So if we move now from this general principle of physiology to what is specific to skeletal physiologies, and again, there are specific features that determine the way it can be approached. The first one is that skeleton is the organ that covers the most surface in the body with skin and muscle. And as a result, if there is an event that occurs in skeleton, it occurs in 227 bones, and the cost of this event is multiplied by the surface covered by this organ. Whatever is the cost, you have to multiply by the surface. The second true and specific particularity of bone is that bone is the only organ that contains a cell type whose only function is to destroy the host tissue. So in essence, bone is the only organ that goes throughout its life through a physiological autoimmune disease. And this cell type is the osteoclast, whose only function is to destroy bone. It does not do it at random. It does it in a physiological function that is called bone modeling during growth, bone remodeling after puberty, in which there is first destruction of bone by the osteoclast, followed by de novo formation by the osteoblast. Not only it occurs every day, but it occurs in 100 locations every day. And therefore, that has many implications. So we all know about bone remodeling because it is a function that is affected in the most frequent bone disease, which is osteoporosis. And by all accounts, it's certainly an interesting disease, but it is not a glamorous disease. It's not a disease that kills anymore. And it's a silent disease, and it's not a disease that is very attractive compared to autoimmune disease. And if we don't think about it, by extension, if the disease is not that interesting, then the function is not interesting. And this is where I think we need to take a step back, because if evolution has kept, like it has done, bone remodeling for 3 million years, it's not just to send older and wealthier lady to the endocrinologist at the end of the 20th century and the beginning of this one. In fact, the true and the original function of bone remodeling is to be an ambulatory orthopedic surgeon. It is here to repair micro and macro damage. It is here to repair fractures. And it is not a function that was existing only in the dark age of humanity, but I bet that in the middle of the 19th century in Bethesda, Maryland, there was not many orthopedic surgeons besides bone remodeling itself. So, as such, bone remodeling is a true survival function, because if you cannot repair fracture, you lose mobility, and that's a problem in some emergency function. And as such, and in objective terms, not in subjective terms, bone remodeling was more important than the control of appetite. Things have changed. We don't need bone remodeling anymore. We would like to control appetite. But during 3 million years, it was much more important. So in biology is like life. There is nothing like a free lunch. And there is a cost to commit suicide, like bone does every day in 100 locations. This cost is increased by the number of times it occurs and by the fact that after that you have to form de novo bone, and this cost is energy. There is no way that the osteoclast and the osteoblast can kill and form bone if they don't have the necessary energy coming to bone to allow this process to occur. So that is a completely theoretical view of bone remodeling, and it can be discarded as French impressionism, but there are clinical observations that support it. And one is that osteoporosis, as you know, invariably follows gonadal failure, and the other one is that obesity protects from osteoporosis. And so clinical observations are like statements of the Old Testament. They can be interpreted in hundreds of ways. They are all right and all wrong, and we chose one in the light of our view of bone remodeling, and we chose it because it allows us to formulate a hypothesis, and this is a hypothesis and not a speculation because it can be tested. The hypothesis is that bone mass, appetite, and reproduction are controlled by the same hormones. And the reason why it is an hypothesis is that we now can, through mouse genetics, test it in a cell-specific manner and in a way that is uncolored and unbiased by any pre-existing theory. You inactivate the gene and you do or you don't have the phenotype. But with mice, there is nothing in the middle. It's also a hypothesis because it is easy to test for appetite. It is easy to test for reproduction or sterility, and it is not that difficult to test for bone mass. So that's a good part of this hypothesis, but this hypothesis has its own logic, and therefore it has its own constraints. So we view bone remodeling as a fantastic change during the evolution, and in fact the appearance of skeleton has completely changed the physiology of animals because of the energetic cost of bone remodeling. Therefore, if there is an hormone that regulates bone mass and energy metabolism, it should appear during evolution with bone and not with energy metabolism, because evolution is like developmental biology. You come up with a gene when you need it, not after. The second thing is that the control of bone mass should have a central component, if not completely central, because the control of appetite and reproduction is by and large of central nature, and physiology is simple. Molecular biology is complicated, but not physiology. And the third one is that what goes around comes around in endocrinology, and if there is an hormone made in endocrine cells regulating bone mass, then there must be an hormone made by bone cells regulating energy metabolism. I'm not going to speak about it today for the sake of the logic of the talk, but this is the work of osteocalcin demonstrating that bone is an endocrine regulator of energy metabolism. So with this string attached to this hypothesis, there are not many hormones that you can test. And there is in fact only one, and it's a very attractive one. It's leptin. Leptin, as you know, is an adipocyte-derived hormone that was identified and put in the New York Times for its ability to regulate appetite. Not body weight, appetite. Mice become obese and patients become obese because they eat more, but the function is the regulation of appetite. In other words, in absence of leptin, you don't need to put mice on a high-fat diet to see them become obese. And it also regulates reproduction. But consistent with the hypothesis, leptin does not appear during evolution with appetite. It does not appear during evolution with reproduction. C. elegans can eat more or less. Flies can mate or not breed or not mate. In fact, leptin appears during evolution with the osteoclast. with the ability of bone to remodel. And before that, in animals that have a cartilaginous skeleton, there is no leptin, much less in fly and in C. elegans. And consistent again with the restriction of the hypothesis, leptin signal in the brain to fulfill or to regulate appetite and reproduction. And therefore, we wanted to test if it was a regulator of bone mass. I'm not going to show the past work, I'm just going to summarize it. Leptin is a powerful inhibitor of bone mass accrual. And by that, what I mean is that mice that have no leptin are menopaused, they are sterile, and have high bone mass. And I don't know of any other mouse model where there is a coexistence of hypogonadism and high bone mass. And it is certainly much more difficult to have a high bone mass when you don't have gonads than to become obese when you eat too much. And that really echoes the fact that leptin appears during evolution with bone. This function occurs only through a central relay, at least in vivo, and it is not a consequence of its regulation of energy metabolism. And over the years, this obviously notion that the brain controls bone was not necessarily accepted immediately. What we decided to do was first to show the nature of the connection between leptin signaling in the brain and the osteoblast. And what we showed is that there are in fact two mediators linking central leptin signaling to bone mass. One is the sympathetic nervous system acting through the beta-2 receptor present on osteoblast, and I will come back to it. And this, in fact, recruits the molecular clock to decrease bone formation and to increase bone resorption. As a result, women that are on beta blockers are protected from osteoporosis because they have a decrease in bone resorption and an increase in bone formation. And then there is another mediator, which is CART, whose function is to inhibit bone resorption. I'm not going to speak about today, but I will use the sympathetic tone as a readout of leptin signaling in the brain several times. So that is what I call the peripheral question about leptin and the regulation of bone mass. But there are central questions, and I thought that in fact this central question had been solved, and in fact we did contribute to some of the experiment, and they were not. And these central questions I'm going to speak about is, where does leptin signal in the brain to regulate bone mass accrual? In other words, is leptin an hormone acting on a receptor in a cell that is responsible of a function, or is it an endocrine modulator acting on another molecule? Where does leptin signal in the brain to regulate appetite? And the third question is equally important. Is there a common molecular basis for the regulation of bone mass and appetite? Because for reasons that can be rationalized but not understood, the control of appetite appears to be much more important than the control of bone mass, although there is nothing more important in biology than anything else. So for the longest time, the hypothalamus has been viewed as a focal point of leptin signaling, and for very good reasons. I will cite the two main reasons. The first one is that a classical experiment in the 40s, when hypothalamic neurons were lesioned with chemicals, rats became obese because they were hyperphagic. This experiment, together with the fact that these two nuclei in the hypothalamus, the VMH nuclei and the arcuate nuclei, are loaded with neurons expressing at high level the leptin receptor, led to the assumption that leptin acts in the hypothalamus to regulate appetite and bone mass. And we certainly contributed to this, at least we thought we did, because when we lesion the VMH neurons in wild-type mice, we obtained mice that had a high bone mass, and that was leptin insensitive. And we concluded that the VMH neurons are anti-osteogenic leptin-sensitive neurons. As you will see, this is true with a twist. Whereas the arcuate neurons that were lesioned with another drug are anorexic-genic, also leptin target neurons. The picture became more complex, apparently, because of this experiment. So when chemical lesioning is achieved to destroy the VMH neurons, we can reproduce the bone phenotype of ObRb mice, of mice lacking leptin. A couple of years later, another investigator, Brad Lowell, did seemingly the right experiment, which is to inactivate the leptin receptor in VMH neurons, and mice have normal bone mass. So there are two ways to look at these two experiments. The most immediate way is to say there is a contradiction between the two, and because genetic is more powerful, is more specific, it's newer, it's probably true, and therefore leptin does not act in VMH neurons to regulate bone mass and in the hypothalamus. And it's possible. However, this statement does not begin to explain how leptin regulates bone mass. Another way to look at these two experiments is to say that it is a blessing in disguise that they were both performed, because they are complementary. And in fact, a very literal reading of this experiment says, and it says only, they say only that leptin cannot regulate bone mass if VMH neurons are not there. But leptin needs not to bind to VMH neurons to regulate bone mass. That's all they say. And by saying that, they immediately formulate a hypothesis, which is that leptin does not act at all in the hypothalamus, but it acts elsewhere in the brain to regulate the synthesis of a neuromediator that then will act in the hypothalamus and will be the true regulator of bone mass, the true central regulator of bone mass. This is a hypothesis we have been working on for the last 4 years. And while working on this hypothesis, we were attracted to one molecule for clinical reasons. And this molecule is serotonin. So let's go straight to the clinical substratum of this work. Serotonin reuptake inhibitors lower bone mass. So serotonin is synthesized exclusively in brainstem neurons, and it accounts for only 5% of serotonin. And serotonin does not cross the blood-brain barrier, and I illustrate that in my lab with new students by telling them that it's a little bit like Las Vegas. What happens in the brain stays in the brain, and what happens with serotonin in the gut stays in the gut, and nothing goes through the blood-brain barrier one way or another. The synthesis of serotonin is initiated by TPH2, therefore mice in the brain. Therefore, mice that have no TPH2 have no serotonin in the brain, and for the sake of clarity, I will call them brain serotonin null mice. So this is to show you just where the gene is expressed, TPH2. We knocked like the allele in it, and you can see that only neurons of the dorsal and ventral raphe are staining blue with sections of the brain from left to right, from top to bottom. There is no other blue cell. And in fact, there is no blue cell in the rest of the animal as far as we can tell. In absence of serotonin, mice have developed an osteoporosis that is quite severe, and it's a dominant phenotype. And brain serotonin, in fact, is a positive regulator of bone mass accrual. It's quite an important finding because since serotonin does not cross the blood-brain barrier, and we verified that, serotonin, or brain serotonin, is the first and only neuromediator that I know of, true neuromediator, that regulates bone mass. So, as such, it is important to understand how it occurs. So, as I will show in the second part of the talk, serotonin made in the gut inhibits bone mass, and it was important to determine which one of the two serotonin exerts a prevailing influence. And to do that, we generated mice that cannot make serotonin anywhere in their body by knocking out TPH2 and TPH1. And these mice that have no serotonin anywhere have the same osteoporosis phenotype that mice that have no serotonin in the brain. To me, that was a complete surprise because brain serotonin is only 5% of total serotonin. But this result has another implication. It implies that the regulator of serotonin synthesis in the brain has a more important physiological in bone remodeling than the regulator of gut serotonin synthesis, and I will come back to it. Because of our interest, or I don't know if we can dim the light a little bit, on VMH neurons regulation of bone mass, we wanted to see if there was axonal connection between brainstem neurons and VMH neurons. And for that, we use a dye that we injected in brainstem neurons and look if it was going to VMH neurons, and it was. And we did the reverse experiment, injected the dye in VMH neurons, and looked if it was going to TPH2 expressing neuron that's stained in blue, and it does. So there are axonal connection between the brainstem and VMH neuron in the hypothalamus, suggesting that, or raising the possibility that serotonin regulates bone mass by acting in the hypothalamus. In the hypothalamus, among the 14 receptors for serotonin, one of them is expressed at a very high level. It's the HTR2C receptor. And by in situ hybridization, we could show that it is expressed in VMH neurons at a much higher level than it is in arcuate neurons that regulate appetite. It's important because mice that do not have this receptor, so that mice that lack this receptor in every cell, develop the same osteoporosis phenotype that mice lacking brain serotonin. And if we do compound heterozygote, mice lacking one copy of serotonin and one copy of the 2C receptors, they also develop the same osteoporosis, indicating that brain serotonin regulates bone mass by acting through this receptor. That did not really address the cell-specific nature of expression of this receptor, and to do that, we use a genetic trick in which the gene, the 2C gene, was removed in all cells and then re-expressed in VMH neurons only. And these mice have a normal bone mass, and that implies necessarily that it is through its expression in VMH neurons that 2C regulates bone mass. So that is for the central serotonin signaling, but at the end of the day, we want to know how the brain speaks to bone and what is the mediator of serotonergic signaling to the bone. And to address that, we were helped by a feature of the phenotype of mice lacking serotonin. I told you earlier that the sympathetic tone inhibits bone formation and increases bone resorption, and mice that have no serotonin have a lower number of osteoblasts and an increase in osteoclast. And they also have an increase in sympathetic activity, measured here by UCP1 expression, and here by epinephrine level in the urine, and again, UCP1 expression. And again, it's a dominant phenotype, suggesting that, in fact, the mediator, the peripheral mediator of serotonin signaling in the brain is the sympathetic tone. And that was proven by removing one allele of the beta-2 adrenergic receptor from mice that have no serotonin in the brain. And even though these mice have no serotonin in the brain, they now have a normal bone mass with a decrease an increase in the number of osteoblasts and in the bone formation rate. The osteoclast number is not changed because of the other regulator I don't have time to speak about, which is SCART. So the way serotonin regulates bone mass, brain serotonin, is by binding to the 2C receptor in the VMH neurons. As a result, it decreases sympathetic tone, which results in an increase in bone mass accrual. And so the question is, what does this has to do with leptin regulation of bone mass? So the fact that serotonin and leptin exerts exactly opposite influence on bone mass accrual, yet by recruiting the same mediator, suggested immediately a model whereby leptin would be an endocrine modulator inhibiting the synthesis of serotonin and thereby decreasing bone mass. So that was tested in many ways. The first one was to show first that the leptin receptor ObRb is expressed or co-expressed with TPH2 in brainstem neurons, and that if we inject leptin in wild-type animals, we get a time-dependent, dose-dependent decrease in serotonin synthesis in the brain. Conversely, if we look now at mice that have no leptin, we can show that already at 6 weeks of age, they have a twofold increase in serotonin content in the brainstem and the hypothalamus, and we can normalize that by removing only one allele of TPH2. So leptin regulates the synthesis of serotonin as expected. It also regulates the secretion of the release of serotonin. And in an electrophysiology experiment, what we did was to take slices of the brainstem and show that the electric activity was decreased when they were treated with leptin and restored when leptin was removed. We can show it specific using, again, genetics. If we use wild-type brainstem slices, leptin inhibits the electric activity. If we take slices in which we have removed the leptin receptor in brainstem neurons, leptin is not active anymore. So leptin regulates both the synthesis, inhibits the synthesis and the release of serotonin. But the proof of principle has to come from genetics, and it has to come from two different experiments. The first one is to demonstrate that mice that have no leptin, but with a normal serotonin content in the brain, have now a normal bone mass. And for that, we removed one allele of TPH2 from ObRb mice, and we normalize the bone mass as well as the sympathetic tone, demonstrating that indeed it is probably the mediator. And we have a converse experiment in which we removed the leptin receptor in various parts of the hypothalamus, the VMH neurons, the arcuate neurons, or in the brainstem. And it is only when we remove leptin signaling in brainstem that mice develop a high bone mass phenotype due to an increase in bone formation and an increase in bone resorption that is again caused by CART. And there is an increase in sympathetic tone. So the two genetic experiments are congruent, and they were based, obviously the last experiment relies on the specificity of the Cre driver that we use. And the Cre driver that we use to delete genes in the brainstem was demonstrated by crossing it with ROSA 26. And you can see that only cells of the brainstem stain blue, whereas cells of the hypothalamus do not. And the way leptin regulates bone mass is by inhibiting serotonin synthesis in the brainstem and therefore blocking the cascade which leads serotonin to bind to VMH and to increase bone mass. Because it does that, it decreases bone mass. So the other central question, as I said, is where does leptin signal in the brain to regulate energy metabolism, and is there a commonality of mechanism? And this question came, and the hypothesis is the same, that leptin does not act in the hypothalamus whatsoever to regulate appetite, but regulates elsewhere the synthesis of a neuromediator that then acts in the hypothalamus. The hypothesis is the same, because the problem is the same. If we lesion the arcuate nuclei with the chemicals, we create an ObRb mouse. But if we delete the leptin receptor in the arcuate neurons, there is no phenotype. Not only the problem is the same, but the suspect is the same, because patients that take SSRI are prone to hyperphagia and obesity. And so we tested it because if it is true, if serotonin is the way leptin regulates appetite, and by the way, serotonin in C. elegans is also promoting appetite, then mice that do not have serotonin in the brain should be anorectic. And they are, whether they are heterozygous or homozygous. The homozygous have a 50% decrease in appetite. They have no fat. They have an increase in energy expenditure. Again, by axon guidance experiment, we could show that there are axonal connections between brainstem neurons and this time the arcuate neurons of the hypothalamus. And in the arcuate neuron of the hypothalamus, there are two serotonin receptors. The HTR1A that is expressed at a higher level than the HTR2B, and POMC is used here as a control of specificity in situ hybridization. When each of these genes, the HTR1A and 2B, were knocked out in a cell-specific manner, we observe a decrease in appetite that was more pronounced in absence of 1A than 2B, for the good reason that 1A is expressed at a higher level than 2B. But again, this is all expression and correlative argument. Again, the proof of principle has to come from the genetic. And what happens if we normalize, not abolish, but normalize serotonin synthesis in ObRb mouse? And what happens is that these mice eat normally. They have no leptin, but they have a normal appetite, normal body weight, normal energy expenditure. They still have serotonin, but they have normal content of serotonin. So the question is, what happens if they don't have serotonin? ObRb mice that don't have serotonin are anorectic. It's not a profound anorexia, but it's severe enough that some of them die of hunger. They don't have leptin, but they don't have serotonin either, and they have an increase in energy expenditure. Conversely, if we use again the same allelic series to inactivate the leptin receptor in VMH neuron, it's a mistake, I apologize for it, arcuate neurons or brainstem neurons, it's only by inactivating the leptin receptor in brainstem neurons that we reproduce the phenotype of the ObRb mice. And it's not only at face value, but if we look at molecular markers now in the hypothalamus, when we delete the leptin receptor in the brainstem, we reproduce the molecular phenotype caused by the absence of leptin. There is a decrease in MC4 and POMC expression, an increase in NPY, and there is also a decrease in the size of the brain and in the diameter of the POMC neurons. So again, the scheme is the same. Leptin regulates appetite by inhibiting serotonin synthesis in the brainstem, serotonin being a positive regulator of appetite. And overall, there is a common regulation of bone mass and energy metabolism. That was an acquisition of vertebrates. Serotonin is in C. elegans, and its function in C. elegans and in human is to increase appetite, and in vertebrates to increase bone mass. What vertebrates acquired is leptin that inhibits serotonin synthesis, and as a result, co-regulate, but in the opposite direction, appetite and bone mass, so that there is a normal amount of energy coming to bone because the two functions are co-regulated in the same direction. And all this work is now on the form of a 4-minute video on the Cell website, which is much simpler than all these slides. So clearly, as I said, there are two serotonins, and even though it's the same molecule, they cannot be the same function because they are separated by the blood-brain barrier. So serotonin in the duodenum is 95% of serotonin, and it has no known function. Its synthesis is initiated by another enzyme, another gene called TPH1. And we were obviously interested in looking at the function of serotonin in the gut, and we were helped in this by true and sheer luck. So there is a human disease that has received a lot of attention, and rightly so, which is called osteoporosis pseudoglioma. And those are kids that are born blind, but with a normal skeleton, and I will come back to it. There is no abnormality of skeletogenesis. And very quickly they develop a very severe osteoporosis postnatally, and it is a lethal disease. This is a tragic disease, and there is another disease that is even rarer and less severe or less tragic. It's high bone mass syndrome. Those patients have no symptom except in 3 occasions. The first one is that if they have to go to the dentist, it's quite challenging. The second one is that they cannot swim because they have so much bone they sink. And the third one is that if they are women and reach menopause, they don't develop osteoporosis because they have high bone mass. And what this says, and it says only that, is that we have in our body a pathway, a signaling pathway, that if we cheat it, protects us from osteoporosis. In other words, we have in our body something that looks like a drug. The two diseases became very important because they are caused by a mutation or different mutation in the same genes, LDL receptor-related protein 5, LRP5, which is a cell surface molecule expressed in essentially all cell types of the body. And loss of function mutation in LRP5 causes osteoporosis pseudoglioma, whereas a single amino acid substitution that is viewed as a gain of function called the high bone mass syndrome. The importance also of this gene is that it regulates only bone formation. Mice that have no LRP5 obviously develop an osteoporosis. They have no bone, the black is the bone, and it's due only to a decrease in bone formation. The number of osteoclasts is not affected. So inhibiting this pathway, or promoting this pathway rather, would mean an anabolic treatment for osteoporosis, which is really the holy grail. So LRP5 is in all likelihood a Wnt co-receptor. It's homolog to LRP6, which is a true Wnt co-receptor. Its forced expression in 3T3 cells increase stop flash, and we have shown that it is as a Wnt co-receptor that it regulates eye vascularization. However, it may not be a Wnt co-receptor for bone, and it was apparent from day one. LRP5 inactivation in mice or in humans does not cause embryonic defect of the skeleton. And that would be the first time that a Wnt mutation has no embryonic defect. The gain of function mutation of LRP5 in the patients, the high bone mass patients, they don't develop tumors. And Wts are oncogenes. As importantly, when we generated an osteoblast-specific deletion of beta-catenin, of Wnt signaling in osteoblast, there was no effect on bone formation. There is an important phenotype, but it's not on bone formation. So none of this argument is enough to say LRP5 does not act as a Wnt co-receptor in bone, but taken together, they whisper that we may want to look elsewhere. And that's what we did. In fact, we went back to square one and tried to understand what is the molecular abnormalities in LRP5 null mice. And the molecular abnormality is a decrease in osteoblast proliferation. And you can appreciate that this decrease in osteoblast proliferation is not present in mice lacking Wnt signaling in osteoblast. This decrease in cell proliferation exists in vivo, but what is quite remarkable is that it does not exist ex vivo. If the cells are put in culture, LRP5 null osteoblast, they proliferate as well as wild-type osteoblast, which indicates that the signaling event responsible of the arrest of osteoblast proliferation in vivo certainly does not come from the osteoblast and may not come at all from bone. And so, based on that, we decided to do a microarray of LRP5 null bones looking for genes that will be of interest. And what we observe is that in absence of LRP5, the gene the most highly expressed in bone was TPH1. And obviously, we were ready for it, not that we were expecting that, because we knew what TPH1 and 2 were. So again, TPH1 is the enzyme that hydroxylates tryptophan and is the first step of a two-step biosynthesis pathway of serotonin. And if we go back now to the mice and to the patients, but to the mice first, if we look at LRP5 null mice at birth, they don't have a bone phenotype and they have normal serotonin in blood. At 2 weeks of age, now they have an increase in circulating serotonin, and things get worse over time, and it is a dominant phenotype, and the LRP5 mutation is a dominant mutation. If we now try to understand why the osteoblast, LRP5 null osteoblasts do not proliferate in ex vivo, if we add serotonin to the culture, we block osteoblast proliferation because we decrease expression of genes cyclin D1, D2, and E1 that are regulated by LRP5. So this was a very strong argument that it was the production of serotonin that was mediating the function of LRP5. If we look now at patients, that is our work, but I know that it has been done in Mike White's lab and labs in Australia and Europe. All the patients we have looked at have an increase in circulating serotonin levels, not necessarily as high as that, but always twofold more than the controls. TPH1 is a duodenum-specific gene, and the level of expression of TPH1 in the duodenum compared to bone is 1,500-fold higher, whether the mice are wild-type or LRP5 null, which immediately raises the hypothesis that osteoporosis, pseudoglioma, and high bone mass were two endocrine diseases of the gut, and serotonin and hormone regulating bone mass, gut-derived serotonin and hormone regulating bone formation. To test this hypothesis, what we did was to do cell-specific gene deletion of LRP5 in osteoblast. It took 1.5 years and a lot of dollars to obtain a wild-type mouse. But when we do this deletion in a gut, we now obtain a mouse that develops an osteoporosis. And remarkably, it does not affect bone resorption. Remarkably also, there is no eye phenotype. And this is consistent with the fact that LRP5 in the eye is a Wnt co-receptor. Conversely, if we delete TPH1 in osteoblast, then mice don't develop a phenotype, but if we do it in the gut, they develop a high bone mass phenotype due to an increase in bone formation, reproducing the high bone mass syndrome. And this phenotype, by the way, is dominant. Heterozygous mice have the same phenotype. I already showed this slide. So the importance of the work on of an LRP5 is not so much as tragic as is a disease, osteoporosis, pseudoglioma, but it's a high bone mass syndrome because these patients have a cure for osteoporosis in their body. So it was important to see that, in fact, LRP5 or serotonin mediates the gain of function mutation of LRP5. And we did exactly the same thing. We knocked in the single amino acid substitution of LRP5 in a cell-specific manner in the gut, and heterozygous mice have twofold more bone than wild-type mice because they have a twofold or threefold increase in the number of osteoblasts and in bone formation rate. No effect on osteoclast. And when this mutation, this knock-in mutation was introduced in osteoblasts, there was no effect. We had access to only 2 high bone mass patients initially. Now we have another family and the results are the same. There is a 40 to 50% decrease in circulating serotonin levels. This is plasma. Previously it was serum. And it is normal that they have serotonin because this disease is dominant. The patients have only one allele mutated. Among the 14 receptors for serotonin present that exist, 3 of them are expressed in osteoblasts, which is the problem. And so we obviously did not know which was mediating the function of serotonin, and for that we did cell-specific gene deletion of each of them in an osteoblast-specific manner. And what we observe is that, luckily, only one of them is mediating the function of serotonin in bone, and it's the HTR1B receptor, whose deletion at the heterozygous state in osteoblast results in a high bone mass phenotype due to an increase in bone formation parameters and no effect on bone resorption. We also could work out the signal transduction cascade downstream of the receptor and show that CREB is the transcription factor downstream of serotonin, and to do that, because the CREB null mice die, we did compound heterozygotes for mice lacking 1 copy of LRP5 and 1 copy of CREB in osteoblast. And these mice reproduce the osteoporosis due to a decreased bone formation phenotype of LRP5 null mice. So what it seems to be is that, in fact, in mice and in human, enterochromaffin cells of the duodenum are a regulator of bone mass. LRP5 and its ligand, which we don't know what it is yet, inhibits serotonin production. And serotonin acts on osteoblast to inhibit CREB function and to decrease osteoblast proliferation. And to the best of my knowledge, I don't know of any other molecule besides serotonin that exerts opposite influence on the same physiological function depending on its sites of synthesis. So this work was done by Vijay Yadav and Frank Couret, and we got mice and patients from many people. And we are funded by the NIDDK and hopefully one day by a PO1 that is on the cusp at, forever at NIA. Thank you very much.
Gerard, so I was curious why the brain serotonin effect is dominant over the gut. You know, the effect must be indirect. And then could you speculate what is that mediate of brain serotonin and why that effect is dominant over the more direct gut-derived serotonin?
Yeah. So one of the assumptions of the work, and the work is not finished, is that leptin is a major regulator of bone remodeling. Leptin is a regulator of serotonin in the brain and should be more important than the regulator of LRP5, the ligand of LRP5. And it's just a rule of physiology that one is more important than the other.
So many other hormones also affect bone formation, including some of the reproductive hormones. So does the effect of leptin affected by the age or the gender? And of course, you studied some of these patients, and does it relate to the effect on leptin as well as serotonin in this age as well as gender effect?
So sex steroid regulates mostly bone resorption, not bone formation. There are very few hormones that regulates bone formation besides leptin and serotonin. The effect of leptin gets worse over time, at least in mice and patients that have no leptin, they get more and more bone. Serotonin circulating level increases at menopause, approximately 30%.
One of your slides indicated that TPH1 was very high, or the only source of it, the only place it was expressed is in the gut. This is really wrong because there's a huge amount expressed in the pineal. So against that background, can you reinterpret your work? Or was your deletion of TPH1 absolutely specific for the gut?
So this is a strongly worded question. This is not wrong in the sense that for every single mouse, we measured serotonin in blood, in the pineal gland, in the brainstem, in the cortex, in the cerebellum, and in the hypothalamus. In absolute terms, when you see TPH1 expression in pineal gland, it looks like it is a lot. But when you do, in fact, compare to duodenum, it is not pathetic, but close to be. Mm-hmm. So I don't think our conclusions are wrong, because in all the mice in which we don't have serotonin in the peripheral circulation, there is absolutely no effect on the brain content of serotonin. And when I say brain, I mean several compartments. Likewise, to tell you how little importance has TPH1 in the pineal gland, in mice that have no TPH2, there is no detectable serotonin in the brain, and there is no effect of this deletion on the circulating level of serotonin. So I will not be as drastic as the way you put it.
Well, the pineal also has the capacity to make compounds that won't be made in the gut. Does that have any influence on your interpretation?
We are not working on the pineal gland. We worked on brainstem and duodenum.
But with the TPH1 knockout,
In the TPH1 knockout, all we looked at was serotonin, and in various localization, and again, there was no effect in the brain, so we did not go further than that.
I want to thank Dr. Korsinski, and I'd like to invite you all to a reception that we'll have for some light refreshments. I believe not usually in the out back, but actually in the library. So we'll have the reception in the library. Thank you.